Okay, so at the end, we usually have a wrap-up session. That's sort of not a new thing. We've done this every time. So obviously this sort of opens the podium for any remarks, questions, suggestions. But and I certainly don't want to inhibit anything you want to say. But I like to perhaps focus it a little bit on, if possible, on two central questions. So the central question would be, number one is, what have we learned about the pathologic mechanism of Barth syndrome? And number two would be, what transpires from what we have learned about potential treatment or management of the disease. Like two remarks from my end, just with respect to the first question. I think at least to me, what has become clearer and clearer and clearer over the years, starting with the zebrafish model that Zaza has created, to our Drosophila model, to the current mouse model, is that Barth syndrome is really a developmental disease. That means it is what the phenotype of Barth syndrome is decided within a few days of intrauterine life, and everything else afterward is just, well, it may modify it a little bit, but this is the critical time that we have to look at. Now, once you have Barth syndrome, what is sort of going on? We've done a lot of work on this. We have heard a lot of good talks. A lot of things are happening, but a lot of things are perhaps not particularly relevant. Mitochondria perhaps are a little bit uncoupled, but for the most part, they're actually functional. But what can Barth mitochondria not do is they cannot respond appropriately to excessive energy demand. So this is when they actually fail. And perhaps this also explains why patients sometimes go into these crises of heart failure. Something must be happening that increases the energy demand. And then all of a sudden they come out of this crisis for no good reason. Perhaps the trigger has gone away. So this is sort of my 50 cents. And but now I want to open the podium to anything you want to say. Yes. Ron?
Yeah, for me, the main message which comes through from this meeting is a bit hinting also to what Michael was saying, that the problems is in Barth syndrome is not so much the respiratory chain, but is a selective usage of the different substrates. It was very clear from yesterday, from Todd's experiments, and also today from Adam and also from Michael, you know. For instance, what Adam was showing, that if you use glutamate as a substrate or malate glutamate as a substrate, then the oxidation was actually increased in Barth syndrome, indicating that the respiratory chain is working perfectly normal as long as the NADH coming from malate and picked up by glutamate is pulled into the respiratory chain. The problem comes in if you introduce the fatty acids. You know, what also Zaza was showing with palmitoyl-carnitine, what you do there, you still have malate as a substrate, and now the oxaloacetate, which is derived from the malate, is picked up by the acetyl-CoA units coming from beta oxidation, and there the problem is. Arnie says it has something to do maybe with the channeling of substrates, that, you know, we think of enzymes sitting in isolation in a kind of pool of water. In reality, all these enzymes are stuck together in a network, and that somewhere there the problem is. But that was something which also from the yeast work, the arginine work, which Richard brought up yesterday morning, and it also came through in the work from Miriam and also from Chris McMasters. The funny thing of this meeting, for me at least, was all these things came together. The yeast work, the work of Richard, and also the work we've heard this morning. Yeah, fascinating actually. And the only thing we have to do is try to resolve what is hidden underneath. But the picture has become quite clear actually during these 2 days. Yeah, that's what I take home.
Well, Yeah, I appreciate that. I agree. I mean, to hear, I thought that to some extent I hadn't even talked to Michael about the glutamate thing until I was here, so I wasn't sure I was going to see anything to support it, and so it was really exciting. And I guess the thing I'd add, and I think it ties the two comments together, and I was kind of corrected. I mean, if you're respiring on glutamate, you really only have one enzyme to produce NADH, and so they're normal, but if pushed, that's not going to do it, right? And so if there's any stress, the heart is just not able to upregulate that system enough to supply energy to meet metabolic demand under any kind of stress. And so it would make sense that it wouldn't be so horrendous and they can survive, but if challenged, it doesn't work. So the question that comes is, assuming aside as something as simple as CoA, which I can't imagine that would just do it, and it hasn't in the past, certainly feeding at least. Could there be dietary interventions or something with Dr. Kelley or someone who is very experienced with the ketogenic diets, the effects of other mitochondrial diseases that seem to have a problem with substrate utilization, where a certain kind of feeding regimen or something we could learn from diseases where we know that's definitely the problem or the phenotype's similar, and is there some kind of a treatment that has worked in those types of environments that could be maybe translated to Barth syndrome if we just theoretically look at it that way?
I hope I'm not repeating myself. I've given so many talks this week. But just to address the question about that Michael raised about the sudden deterioration and the increased need, I don't think it's that strange at all. I've been gathering data on the cardiac parameters of our patients, the ones who suddenly go into heart failure. And most of these had been boys who are going into puberty, and they go into heart failure at that point. And although it's not just those, and what the data show is, especially for the boys in puberty, is that one can trace back the beginning deterioration of the heart 2 years before they suddenly go into failure. And what happens is the heart is gradually dilating. It's typically the values stay within the normal range. And if you look at the cardiologist's report, they say stable. And that's because the ejection fraction is stable. It doesn't change. And the point is that the ejection fraction is the last thing to go. The heart is dilating for again, I believe, a nutritional reason. That you're getting gradual thinning of the myocardium, but because the Z-scores don't go out of the normal range, and because the ejection fraction stays the same, there's usually not any concern. And the heart, of course, has its own natural mechanisms for maintaining contractility. So a lot of these, quote, sudden cardiac deteriorations are not because of a sudden increase in need for something. It's because they've run out of contractility preservation, and the bottom falls out.
I mean, if the heart dilates, the ejection fraction has to go down, because if it doesn't, the stroke volume would go through the roof, right? So, I mean, dilation and cardiac dilation and reduction of the ejection fraction always go together. But I guess the problem is that ejection fraction and dilation are very dynamic parameters that depend on a number of other things, as the filling status of the heart and so on and so forth. So they are notoriously unreliable,
Well, I know it doesn't make sense, but if you look at all these reports, I mean, it does make sense when you look at what is dilating and what is not. But as they say, again and again, the ejection fraction stays constant while the heart is dilating. Just looking at the left ventricular diameters at systole and diastole.
So your feeling is there is a period that leads to that sudden deterioration.
Yes,
that's what you're saying.
All the ones that I've looked at so far, right. I'm sure it's not the only mechanism, but a lot of these, and the important thing is that the cardiologist should know in this particular condition, to pay more attention to the diameters and other parameters than just the shortening fraction.
I think it's important to remember that cardiolipin does so many things that if you think about what Adam was saying, that maybe there are some dietary or other kinds of therapies that might work, I would just like to suggest that it's probably not going to be a single thing that works for everybody, because if you can imagine, if and this also speaks to the fact that there's so much disparity in clinical presentation, that if you're a person that maybe has a different way of utilizing amino acids, or you're particularly strong in utilizing a particular amino acid, then you may not have such a severity in the clinical presentation. So it's just important to keep in mind that any of the functions of cardiolipin has the potential to be a modifier of the phenotype.
Yeah, I think that's a good point. I was asked for the mic before when there was a discussion of echocardiography, and I just, in heart failure, I don't know what the standard of trying to get it to a diagnosis or monitoring children is, but I know just from my understanding of heart failure, looking at things like filling, EA ratio, tissue Doppler, are always going to be very prognostic before you're seeing any real global changes in ventricular function or dilation or any of these things. And so I would hope that would be a part of the series of things that could be measured as well. And I don't know if that's been done or if those are things that have been seen to precede contractile dysfunction, but there's certainly a whole lot more to echocardiography now than just ventricular diameter and fractional shortening, and they could be picked up in flow parameters, Doppler, et cetera.
Arnie, you want to say? You should.
I don't want to defend cardiologists, even though I am one. There are a lot of measurements that could be made, and I think we would want to make, as Lynn Jeffries said, to try to prognosticate. But in fact, I think we all know that both these boys and many, many other types of genetic cardiomyopathy patients can and do deteriorate very suddenly or drop dead. And so I don't think looking at cardiac function is particularly helpful, except to try to measure consistent changes in the models. I'd like to go back to the idea of using the models to test some of the highly speculative theories that have been suggested. Bill Poo's examples, I think, are very helpful of what we need to be able to use the models for, and then to try to extrapolate what those changes actually, why those changes happen, whether it's arginine plus cysteine or some other measure. I do agree with Ron, though. I think one of the big problems that we haven't recognized is these kids are living off of sugar. And they're not living off of fat, at least long-chain fat. And that's a real problem because of the inefficiency of using sugar. So I think we really ought to understand why they can't use fat.
I have a question about, for people who work with tafazine. What puzzles me most from basic science point of view, that when you have an enzyme which is claimed to be transacelase, when you have this enzyme be deficient or knock out, you should not accumulate monolysyl or dilysyl cardiolipin, because that enzyme is not inhibitor of phospholipase. So I wish this kind of question need to be addressed in the future.
Yes,
probably help us to understand better what tafazine is about. Yeah,
I agree with you. And we're actually doing this right now. So this is sort of very preliminary, but it looks like that the accumulation of monolysyl cardiolipin is probably related to the degradation of mitochondria and has nothing to do with the actual remodeling process. So it's sort of, we got fooled into believing for a very long time, of course, because it made sense initially that monolysyl cardiolipin increases because the transacelase that removes it is deficient. But I don't think this is actually the case. I think monolysyl cardiolipin increases because because there is a degradative pathway that's being set into motion. Now, this is a speculation at this point. We don't have the data to really prove that. But we are sort of working towards that.
I think Dr. Zhang's model should be able to, well, unfortunately, in her case, she doesn't have a catalytic synthesis. Let's look at another mitochondrial disease that causes mitochondrial apoptosis to see whether it's the degradation. If that's the case, we should be able to see increased level of the byproduct, right? MLCL and DLCL.
I just have a comment also in relation to what Ron and what Arnie said. I just suggested also to Todd to look at how, instead of looking at exercise and looking, or in addition to looking at exercise and characterizing metabolism through stable isotopes, also looking at fasting. Because if you want to get a grip on how fatty acid oxidation is proceeding, and to characterize how glycogenolysis and gluconeogenesis is occurring, that is maybe something that should be considered to do. These are not nice experiments to do because you have to fast for a very long time, especially when you're an adult. You have to fast about 48 hours. 48. Before you get ketotic. But then you will get an idea if these guys are able to do so and really get fatty acid oxidation on the way.
I think fasting these boys for any period of time would really be contraindicated, but if the mouse model's as good as what's been presented here in the last 2 days, that's what that's for. And
use the mouse
excuse me?
The mouse.
What about it?
I use the fasted instead of you.
Yeah.
So I think this whole question of not being able to meet excessive energy demands, that kind of triggers, I think, two ideas, two questions really. One is, what is it really what you need to make energy efficiently, right? And the second question is then, if we don't have that, then could we just boost mitochondrial biogenesis and at least alleviate some of these symptoms? I mean, that's obviously something that Min-Nong is trying to do. We will see whether this really pans out, but that's at least, it's not a particularly ingenious approach, but it is at least some that may improve things a little bit. Now, regarding so what do we really need to make energy efficiently? So, and I just thought about this now. If you sort of look into different organisms, different tissues, whenever you have high energy efficiency or high energy turnover, you get almost crystalline structured mitochondria. You get an enormous amount of regularity in the overall structural arrangement of mitochondria, right? And this leads sort of to a new era in the understanding, I think, of mitochondrial morphology, which is the three-dimensional long-range molecular interactions in the crystal membrane. So we think of the membrane as some sort of a bilayer that's sort of in which these proteins and now, as we know, protein complexes are floating around. But I think this is probably not true. There is a fair amount of long-range order in that system. It's just very difficult to exactly pinpoint what this order is because it's somewhat flexible, right? I mean, it can be disturbed, but they are not distributed randomly, right? I mean, that becomes clearer and clearer. So something must be going on in Barth syndrome that disturbs that long-range molecular order. Zaza?
I just want to comment on Fred's comment about fasting. We know that in the fatty acid oxidation deficient mice, fasting really precipitates the phenotype, thermogenesis, and tolerance to the cold. Therefore, our mice, Tafazine mice, we also fasted before experiment. We haven't tried to do that with the food in the cold, but those data reduced energy expenditure in tafazine mice were done on fasted mice. They were fasted about 16, 18 hours the day before, so we haven't tried. We didn't go yet. We didn't, went so far to 48 hours, but it's certainly worth to do. And another thing, we should recognize that mice we keep in our vivarium, they are at 22 degrees Celsius. They are not physiological thermoneutral conditions, and there have been lots of papers in the last year, that you need to keep mice at 30 degrees to unveil all the phenotype, and that's the question was raised, or the pharmacological companies, they test mice at 22 degrees instead of 30 degrees. 30 degrees is not very convenient for experiment, because mice, it's very hard to handle mice. They become very aggressive, but we can see the more phenotype on these mice if we keep them at 30 degrees, because heart rate will go from 400 to 600 to 800. And that can be another level of stress. Yes, we will probably do that, but it will be interesting. I don't know what to expect.
Yeah, 30 degrees C is not good for the investigator either. But
right, that's why we have an environmental chamber where mice are, that we outside.
A glove box? I thought you were a serious mouse person, Zsa. I remember going in the cold room with a hat on just to isolate enzymes, right? I mean, you have to be dedicated, right? What questions, please? Comments, insights?
So I have a question, sort of following Dr. Strauss' comments, and so it's just for my own clarity. So in the Barth syndrome patients, is there any fatty acid oxidation defect, or is that known, or?
Can you repeat?
Right, so the fatty acid in the Barth syndrome. So in the Barth syndrome patient, is there any fatty acid oxidation defect?
Well, I cannot speak for, about patients, because I don't have any cell lines from patients. I only can do the comment on the mouse. Yes, they are defect when we switch to the stress. When we apply to high workload, then this fatty acid oxidation deficiency becomes apparent in mouse model.
From a clinical standpoint, the evidence that there's a fatty acid oxidation problem, it wouldn't classify as a defect in the sense of a beta oxidation defect, is shown in the obvious failure of utilization of the fat. I know that I have the experience of seeing a few boys in the newborn period who came in quite ill, and they had moderate ketones in the urine, so at least their liver can oxidize fat to some degree. But there's no sign of a specific block. So my assumption is it's simply there is a mismatch between the amount of fat that is taken in and the amount of fat that is burned in a great many of the boys. So if you want to call that a fatty acid oxidation defect, then it is. And my assumption, or the way I would interpret it, is simply that this type 1 muscle is not working very well. There's very little oxygen uptake. So there's very little need for fatty acid in muscle, which is the major part of fatty acid utilization in the diet.
Based on our model of the ALCAT knockout mice and overexpression, in the overexpression cell line, the fatty acid oxidation is completely impaired. And if you look at the TFP, which is the trifunctional protein expression level, it dwindles. In the knockout mice, where it's completely reversed, you see improved fatty acid oxidation. And that's why I remain optimistic, even though my theory about DHA may need further approval, more data. But nevertheless, I worked for a drug company for over 11 years before I come back to academia. My primary focus is to work on genes that the information we produce from the lab would be useful to guide the drug development. That narrow the research we do in the lab, mainly focus on proteins and enzymes that are so-called druggable. So let's see how you treat Barth syndrome. You cannot give them oxidant. That would not solve the problem. Their mitochondrial defect is the main defect. What can reverse that defect? I mean, so far, the only information I know of that when we knock out this enzyme, it reverses whatever the defect the Barth syndrome has. We are crossing our mice with the Barth, the test-inducible mice with the help of Zaza. And we will see very soon whether ALK deficiency would reverse the defect in the tyrosine deficient heart. I remain hopeful because everything we see so far is if that's true, all we need is inhibitor. You cannot develop activator of transcription factor or kinase. Those are far-fetched. The only thing you could do is to develop, G protein coupled receptor inhibitors or enzyme inhibitors, some extent to kinase inhibitors. Any other protein structures, whatever, they are not even paid attention by drug company.
The other, I may have mentioned this, but it won't hurt to re-mention it. But my thinking about the deficiency of cardiolipin is that if that has been thought and has been shown to be such an essential lipid, there must be something taking its place. And the investigation, this kind of investigation with other enzymes that modify or alter the remodeling of cardiolipin could be very important. That could be the genetic basis of why some boys do remarkably better than others. And that there could be, again, depending on what that lipid is, there could be a dietary effect. Or it could be modified by diet or by some other mechanism to induce the production of the alternate lipid, phospholipid, that is taking the place of cardiolipin.
Just think, one thing in being asked things that we were surprised to hear, I think that were important about the meeting. I think that the talk given by the woman in front of me, I can't remember her name. The evidence of cardiomyopathy and Barth syndrome diagnosis with normal cardiolipin and no neutropenia, right? So two very hallmark parts of this syndrome. At least I thought so. I presume everyone as well. And so in looking at the differences and similarities, as far as we can go with it between those boys and others that have the cardiomyopathy, I think will help to figure out what about the syndrome is really key in terms of the cardiomyopathy, because I mean that was the commonality, right? They had this, but not the cardiomyopathy, and so it gets at there might be something else, and so if it's the amino acid profile in the serum, or if it's something else.
Because the tafazine may be normal. I mean, the tafazine may be functional, so there might be another mutation, of course. So anyway, there's a lot of questions there. Yeah.
I mean, this two boy could be just a normal genetic variation of any gene in polymorphism. I mean, the substitution may not change a thing in terms of functional wise.
Well, in response to Richard's comment, in yeast, if you knock out the synthesis of PE in the mitochondria, you exacerbate the cardiolipin defect. So it could be that PE is, a potential lipid that could compensate. Because they're both non-bilayer tending lipids.
So my perspective on this is that we still have a cardiolipin defect in terms of molecular species diversification, the content of cardiolipin reduction, and accumulation of a monolysic cardiolipin in a tafazine deficient situation. So that's really complicated the problem, and we don't know what exactly is really causing the symptom. And so it would be helpful if we have something like in yeast, like a cardiolipin synthesis knockdown model in mice. So that would clear things up a little bit, because there you wouldn't have a monolysyl cardiolipin accumulation, but you only have a reduction in the cardiolipin level. Then the comparison of the phenotype, and you may find out what's really causing the problem there.
We had the same idea. So the approach that we're in the beginning of taking is to do a PGS1 knockdown, because if you actually deplete CRD, even if you inducibly deplete it, you accumulate PG, which is obviously just another variable. So we're thinking, and we're in the process of getting an inducible phosphatidylglycerol synthase, and then once we can show that we can inducibly regulate the levels of cardiolipin through this, then combine that with the CLD1 knockout. So then we can ask the question, is it the total amount of cardiolipin? Is it the acyl chain composition? Is it just the depleted amount of cardiolipin? And then compare that to either a delta TAZ strain, which has elevated monolysyl cardiolipin, or better yet, we've tried to overexpress CLD to see if we can further deplete the steady state levels of cardiolipin, but that didn't work, and our thinking is that, as you can see, maybe saw from the lipidomic data, I think there's some newly synthesized cardiolipin that is not a substrate of this enzyme, or even we actually tried to express IPLA2 gamma, so maybe the mammalian enzyme would have a different substrate specificity and can drive down what the remaining amount of cardiolipin. Apparently, it's not really the lipase that initiates remodeling in mammals, which is something that's great. So we can try maybe the B isoform, which you guys showed is working in Drosophila. But yeah, that's the goal, to use to really get at that issue.
There are, of course, cardiolipin synthase knockdown or knockout models, like in the fly, for instance, right? We have it, so. And we are, of course, very sick. But in the mouse, I think it doesn't work, right? Michael, did you do this? The mice cardiolipin synthase knockout is lethal, right?
We didn't do that.
Never tried that, yeah.
Because we probably assumed it would be lethal.
Right. The flies are very sick. I mean, that's all I can say. That it's actually visible, In contrast to tafazin knockout, which you need some tests to establish sickness. Yeah?
Okay. I have two very, very simple questions. First one, when we pick up the heart cells, just like a cell line, right? And no matter which kind of cell line from which kind of species, always L4 cardiolipin, just tetra, linoleic acyl cardiolipin decrease. So other things goes up, just like more saturated cardiolipin going up in cell line. Never same as in vivo heart cardiolipin. In vivo heart cardiolipin, no matter mammalian cell, mammalian tissues, or mice, rat, or bovine, or human, always L4 cardiolipin is dominant. But when you make the cell line from them, they change. L4 CL never dominant. So I think maybe some function we really don't know for L4 cardiolipin, because L4 cardiolipin dominant in heart. Because heart problem is a dominant problem for Barth patient. We really don't know. So because our Barth models not so precise, as humans. So we can find a lot of pathological characteristics, a lot of things, but you couldn't find out why function bad. We don't know. Why L4 cardiolipin is so critical. We can change. For Barth patient, more saturated fatty acid cardiolipin there, they should be better to resist RAS. But why their heart function not very good. This is first very simple question. I really don't know. Second one, for Barth patient cells, recently experiment I found same as yeast cells. When for Barth patient lymphoblast, PE increase, cardiolipin decrease. So PE increase very high level. So I think PE maybe compensate cardiolipin function in any tissue or cells outside the heart, maybe outside muscle, I don't know. So this is why yeast model, still very good model, I think. At least for PE, why PE works to compensate cardiolipin deficiency.
Well, maybe, I guess our hypothesis is PE works because maybe some of the properties of cardiolipin, some of the effects of a cardiolipin knockout have to do with non-bilayer formation. So cardiolipin and PE are non-bilayer forming lipids. I mean, that's a big stretch to go from there to why it works. But I think that I just want to also pick up on Steve's point and maybe a little bit on yours, that we really don't know if the defect is because of the decrease in cardiolipin or the increase in monolysocardiolipin or the aberrant cardiolipin chains. I mean, there's no, and nobody even talked about that. I mean, we just don't know that.
Well, and there's a third or fourth possibility, which is that the actual composition doesn't matter at all, but it's the process of remodeling itself that when eventually generates a healthy Quiste membrane, right? And if this process is not there, then the Quiste membrane is not healthy, but the composition is just a side product of this actual remodeling process, which, just is there in order to produce normal assembly.
One thing I took away from a lot of mitochondrial talks is the dynamic nature of mitochondria fusing and fissioning and moving within the cytoskeleton of the cell. And it looks to me that the mutations, those subtle, must affect those very complicated process, and the end result is some complicated phenotype that we see. So I'm just amazed by the mitochondria's effect on so many different things, which is maybe what we should have expected, but not what we were taught in graduate school, that it's just the energy producing part of the cell. It's very, very fascinating, its complication.
But on the specific, on the topic of fusion, it might be even simpler than that, because Jody Nunari's lab showed that cardiolipin is specifically required for for the what's the protein? Whatever. For one of the one specific protein. No, it's MGM. It's MGM. It's MGM assembly. Yeah. And so if you knock out and then if you knock out cardiolipin and PSD1, you don't get assembly of that complex. So that part is actually pretty simple. Maybe not so simple, but
I won't be surprised. I won't be surprised that all the regulator involving fusion and fission require mitochondria phospholipid like cardiolipin, because this it's natural that during the remodeling process it required the quality control as well. That's mitochondria fusion fission is doing. I have actually answered to your question why the cell line different from the tissue. Well, we all know that all the cell line we have so far, they are not human tissue. They are cancer cell line, and cancer cells actually screwed up the cardiolipin profile completely. If you look at any of the cell line, even C2C12, H9C2, we work typically in our lab to try to mimic when's the cardiomyocyte, when's the skeletal myocyte. Their cardiolipin profile are a mess. They are not dominated with tetralinoleal cardiolipin at all because they are cancer.
No. Yeah. But the reason that cancer cells do that is because they rely heavily on own fatty acid synthesis. So they make they make monounsaturated fatty acids and saturated fatty acids, incorporate that into their cardiolipin, and they don't take a lot of fatty acids up from the medium, and that's why they do that. And that's also because they grow a lot faster. They also use glutamine. And they also use glutamine, and that also feeds into the fatty acid seed. Yeah, there's maybe some relation there, but I think that's what. Glucose. Yeah, that's the reason why.
Yeah, and also the predominant fatty acids in the calf serum or whatever you're using are going to be oleate and palmitate. So that's what's going to be taken up. That is one reason. The other reason is that you're converting more to a fetal phenotype. And say if I just analyzed some 2-day-old rat heart, and that is primarily oleate in the cardiolipin. So that's another reason. But I had a comment and I forgot it. So.
One thing I particularly noticed, there's a lot of data being accumulated, lipidomic, microarray data, and there doesn't seem to be a consensus of what it's actually outputting always as a phenotype. And one of the reasons I actually switched from academia to industry was because I was always trying to figure out what cardiolipin is doing, but by doing these connective experiments, sometimes you can never really connect the dots. And the company I'm working for does these Bayesian models of multiple omic data to try to actually figure out the pathological causes for, whether it's a proteomic cause, where they really shouldn't just be focusing on cardiolipin, or maybe it's a transcriptional network. So, I'd be very interested in collaborating down the road to integrate it, to design the right experiments to figure out dynamically what is leading to heart failure. Because I don't want to have to keep chasing around the cardiolipin tail, to figure out what could be actually the causative effect, whether it's bioenergetic or lipidomic, actually figuring out the root cause. Because I think it's probably embedded in a lot of the research and data being generated, but we probably haven't just made the connections together yet.
So I just have a wouldn't call it a suggestion. So actually I went to a mitochondrial fission and fusion meeting last year. So I talked to a few people, and Heidi McBride and David Chan and Judy Naari. So they all seemed really interested in cardiolipin. They sort of asked me questions about cardiolipin, and I don't know that much. So I wasn't sure if the committee interested in inviting those people to be part of this meeting in the future, and so that can help both to getting know better about this.
I remember my little question to the mouse people. So for those of you who've done the mouse, induced it with doxycycline and then taken it away, has anybody done EM to see if the mitochondria is in the proper position for recovery? Okay.
Yeah. So I'm interested in working with the embryonic stem cell, not the MEF cell, the mouse embryonic fibroblast. Anybody did inducible using culture of the MEF from the tafazine transgenic? Because that should be a good model to work with primary, culture of the cell. You throw in the, dox, induce, in the culture. And that will allow you to monitor mitochondrial behavior far better than isolating, cardiomyocyte, because they don't culture long.
Good suggestion.
Good.
I think one of the things that we also have to consider is the rapid development of personalized medicine, and that with the advancements that they are making now, that eventually, prenatal screening is going to be perfect. So we'll be able to catch Barth mutations very early on. And with all the information that we received this week about neonatal effects, prenatal effects in Barth syndrome on cardiac deficiency, trabeculation, etc., I think we have to, as a foundation, position ourselves in a way in which we can be able to treat the prenatal aspect of this. So that might be an area that the foundation would consider focusing on.
Okay, I think we have reached the point of exhaustion.
Yeah, pretty much.
I mean, I don't want to cut off the discussion, of course, if there's anything. Yes?
Hi. So just maybe two questions, a boring question, maybe a more interesting one. One is the interesting one. If this is a development, I come from outside of the field, of course. If this is a developmental disease to some extent, so does that mean that there is a step in the development of normal heart where the metabolism has, everybody now seems to agree, involving glutamine consumption or arginine consumption via alpha-ketoglutarate and other modifications related to the Krebs cycle or whatever? Is there a step in development where this really occurs, maybe to a lesser extent, which is exacerbated in the Barth syndrome because of some kind of arrest at a developmental stage of that kind? And so is it possible to study this process maybe with IPS or something like that? And then in order to see whether it is a compensation, just a pathological response to a pathology, or just the exaggeration of something normal. Normal at some point of development. So what to do exactly to study this developmental aspect? And then the other thing is that I've heard of many possible therapies, partial therapies maybe, from arginine to resveratrol, which looked like they were working in some, experimental models and maybe not others. Is anyone going to try to combine these therapies a little bit more than I have seen here? Because I've seen, testing one or the other, a few of them separately, and try to combine them to see, in vitro or in an experimental mouse model, whether they work better together and whether they would help in many different, stress conditions.
Well, to answer some part of the second question, that is a topic for our scientific and medical advisory board come tomorrow morning to discuss and to evaluate and to continue to do that. So to answer the second part of your question, yes, and there is no company who has a project that's designed to find a drug for Barth syndrome. This is it, though. So in the lapse of that, we will do the best we can, and I think we'll do really well. And we'll pursue with the best minds that are assembled here and ask for help wherever we need it. So that's a short answer to your second question. And the first question, I can't remember now. Oh, IPS cells. I am sure, since I know Bill Pooh talked about IPS cells. Unfortunately, I wasn't here. There's at least one IPS cell line available from a Barth boy. So there's going to probably be others. There's a tool to work with. So no question that that's important and can be pursued. So
tomorrow the SMAB meeting is going to be starting 1 hour earlier. I managed to convince Matt. So 7:30 we'll start with breakfast.
Anesthesiologist hours. So
that would be 5:30.
Okay. Before everybody goes, make sure you fill out the survey. And I'd like to wrap up with what I think is a very important point. I want to thank everybody who came, contributed, talked, and just, I think it was a superb meeting. And I am hoping that there's many collaborations that are been invigorated and been formed in this thing, and I think they have, and new ideas have been generated. But I'd like to close with a little bit of Ben Thorpe, as a boy, Barth syndrome, who introduced one of these sessions 2 years ago, and unfortunately he's no longer with us. But I think part of his introductory talk has a lot to say for us today, and and forever until we find something important. So, Kendall, could you hit it?
When a Barth sufferer gets sick, it doesn't only affect the Barth sufferer, but it affects all his family and friends. So by finding a cure, you won't only be helping the Barth boys, but you'll be helping a lot other people of the Barth boy who knows him. When Michael Bowen Jr. died, the Barth syndrome world came to a temporary standstill, as with Michael Telley's and Jamal. Please give us treatments to try, and if they don't work, we'll try again. We are one big family just trying to find a cure, and if we work together, maybe it'll come quicker. Please don't let us lose any more of our family members. Thank you for this. Thank you for all the hard work you doctors and scientists are doing, and thank you for this opportunity for me to talk to you today.
Thank you, Ben. So with that, I'd like to thank everybody. And remember, there is a social tonight starting at 7 p.m.